pleasure to be here and to tell you a bit more about the current status in medicine. I'm in the unusual situations that I seem to have to defend personalized medicine. There are parts of medicine which are truly personalized, and I'll show you the differences between those which are and which are not. The basic problem we have is that we are all different. A fact which has been widely ignored in large parts of medicine and pharma development. No tumor has ever been seen or treated before, and they are heterogeneous, as you have heard before. So if you break your arm in a complex break, you get a truly personalized treatment because the doctor will use imaging techniques to identify the exact situation in your arm, and from experience knows how different ways of treating the situation will affect the outcome. So he has both the data he needs to personalize the treatment and the experience and the prediction power to do it. If you get a drug, something completely different happens to you. Basically, it's the equivalent of having your arm broken and then in the left arm broken and then in the hospital getting a cast on the right arm because the doctor cannot distinguish the situation, and in a clinical trial maybe there were more people with a broken right arm, and that's because the doctor has no access to the molecular networks, information on the molecular networks, which also differ from patient to patient, and he doesn't have the prediction power to translate this enormous amount of information into predictions of the complex changes introduced by drugs with complex mechanisms. So it's a purely technical point which discriminates between areas in which we have truly personalized medicine and areas which we don't. In drug development, we have at the moment neither the information, neither the data, nor the prediction power. Now, therefore, we have that situation. Surgery is truly personalized. Drug therapy based on first-line treatment is not. Biomarkers or precision medicine are stratified, and the flagship project which we have proposed is will be hopefully truly personalized. That has all sorts of consequences. In 2015 there was a paper in Nature which argued that for the 10 highest grossing drugs in the US in that year, the response rates are represented by the blue figures in that diagram, and you can see there are a lot more red figures, non-responders than responders, with a response rate ranging from between 4% and 25% according to this particular analysis. This is not the only problem. Drugs can cause adverse side effects. In Europe alone, drugs lead to more deaths than colon cancer, just behind the deaths due to lung cancer, the more lethal form of cancer. This increases enormously the healthcare cost. We spend in Europe roughly 4.5 billion euros every day on healthcare. Compared to that, many of the problems which get the European people excited, like the Greek debt crisis, are really small fish. Simply 10% of that roughly goes for drugs which do not work on the patients. Yeah? Okay. I think I thought I was talking into 2 microphones already. Okay. They are going back. We have 4.5 billion euros in healthcare costs. I don't obviously know the numbers for Israel. Roughly 10% of that simply are the costs of drugs which do not act positively on the patients who receive them. Probably another 10% are the medical consequences of treatment with those drugs. 5% of all hospitalizations, for example, in Europe are due to adverse drug effects. 5% of the deaths in hospitals are due to adverse drug effects. And probably a truly personalized prevention could probably save another 30% or so. So I would argue that we could, in principle, save of the order of 2 billion per day if we just did things right in medicine. These costs are not stable. They increase faster than the gross national product, which even if you are not an economist, you will realize are a complete disaster, even in societies with a stable population, and most of Europe is aging, so it is obviously a situation in which we are essentially rearranging deck chairs on the Titanic and not very sufficiently about icebergs. Cancer is a particular bad example in a sense, or under other respects a good example. In some cancer forms we have clear increases in survival time. In other cancer forms we don't, by and large. You should not forget that early detection obviously simply increases the survival time by itself, and obviously also makes the cancers easier to treat. So some of the progress is simply due to the screening approaches. Now, the first part of the problem I've raised, the data, we have a much better grip on. My department was involved in the Human Genome Project. We were centrally involved, for example, in sequencing chromosome 21, the second genome, the second chromosome, which got published. Any luck? Yeah, that's much better. Okay. Okay. So at that point, we used machines which were, in the end, able to sequence 196 independent sequences in one go per run. Now we sequence billions. We sequenced one genome in 13 years for roughly $3 billion. Now we sequence 60 on a single machine run for roughly 600 or more dollars on the way. We are on the way to the $100 genome. So we have enormous progress in characterizing individuals, individual patients. We cannot learn more in a sense of an individual patient than we knew about all of biology, human biology a few decades ago. This is not the only technique which is moving rapidly. This is, for example, a picture from a so-called CyTOF, spatially resolved CyTOF experiment showing a tumor in which you can detect up to 40 proteins with cellular resolution, characterizing the microenvironment of tumors and the heterogeneity of the tumor in much more details than possible up to now. And there are many other technological breakthroughs which we can take advantage of in characterizing every individual patient. These techniques have been used extensively to characterize tumor samples. 35,000 cancer genomes have been sequenced already, and in many cases this can point out actionable variants, which then lead to the patient getting a specific drug or being included in a trial with specific drugs. We carry out both in the department for research grants and in a spin-out company, Alagris Theranostics, which we set up to really make the technology available. on a much broader basis. Max Planck has many positive features, but it's not a place where you can start changing the world. So for that, we have to have other structures to carry out an extremely deep analysis of tumor patients. This sequencing has been used at different levels of accuracy, from individual variants, just basically determining the different RAS mutations, over cancer panels, over whole exome, to what I consider the minimum you really should do on every patient, which is low coverage genome to identify copy number variation, LOH regions. A deep exome has to be deep because the tumor material is not necessarily high purity. It contains, the tumor is heterogeneous. And a deep transcriptome, because obviously it's as easy to stop recessive oncogene from acting by methylating the promoter as by finding, as by introducing a mutation in the gene. So if you just sequence the genome, even if you do a whole genome sequencing, you have probably only half the information you need to really decide on the appropriate treatment of the patient. This type of information is then translated into a so-called CMTA report for the molecular tumor board at the Charité, and goes into the treatment decision of the different patients. Both within research projects, we are running basically a sort of clinical trial on metastatic melanoma, trying to test the success rates of treatment based on the results which we generate molecularly, and those are some of the patients we've analyzed. And as in other centers, you get sometimes dramatic improvements in the status of the patients with metastasis melting away, but you also have the problems that they very often return due to the heterogeneity of the tumors. So we have precision medicine tools which now allow us to identify drugs in situations where we have a high degree of causality. Particular situation where we have a drug, for example, like Lehrach, made against a fusion protein, which we can characterize molecular by, for example, the transcriptome sequencing. The other high causality observations, but in many cases, this is still in the best case only, help a fairly small fraction of the cancer patient. To go beyond that and to go from the stratification which we have in precision medicine, where we have very precise data but a very imprecise predictions typically, to a truly personalized therapy and prevention choice, we think we will have to take an example from what has worked in all other areas of our existence. In all areas of our existence, except for medicine and drug development, we have learned that in complex situations we cannot avoid making mistakes, but we can make them without consequence, safely, quickly, cheaply, on computer models of the real situations. So we don't design cars, testing them on prototypes. We don't train pilots on planes full of passengers. We have computer models, flight simulators, virtual crash tests. Even developing electric toothbrush wouldn't be done in reality. You would develop it first as a computer model before you start actually doing something. It's only medicine in which we really gamble with the lives of the patients which are getting treated. And this is obviously something which has been impossible to change for a long time. We just didn't have the data to make any models of that type, because data are are the key to any forecast model. That's a quote from a meteorology paper. Models are only as good as the data you could put into the model, the data you need to initialize such a model. In weather forecast, we have roughly 75 terabytes of information to initialize the models for every day's weather forecast. Far cry from the doctor having the blood pressure of the patient. If we had the equivalent information on a weather model, which we have typically in clinical practice, the weather forecasts would be barely able to decide between summer and winter, much less get good prediction. And just as a side effect, because I keep getting that question, the weather is a much more difficult problem in some sense because it's chaotic. So very small changes in the starting conditions translate into enormous effects after 3 weeks. That's not the case in biology, otherwise we wouldn't be here, and otherwise identical twins wouldn't look similar after 80 years. So you don't have to worry about that. We have an easier job in a sense. Obviously, the models which we have to build are different. Partial differential equations do the job for the cars or the weather forecast. In simulating biological processes, we use object-oriented modeling systems, which then translate automatically translated into systems of differential equations. I'm convinced that basically differential equations are appropriate technology for modeling biological processes. If you would need stochastic differential equations, we again wouldn't be here to discuss it. So the object simply corresponds to the components of biological processes. You have an object in the model of a normal cell corresponding to the RAS protein, and you might have in a particular model of a particular tumor, in addition, object which corresponds to the mutated RAS protein, which has then changed functions. For example, it will activate downstream processes irrespective of upstream stimulations. Those models can then be automatically translated in systems of differential equations, and the systems of differential equations reflect the accumulated knowledge from basic research over decades, probably funded by $1 billion or more of research funding all over the world. So we can probably rely on the fact that the topology of the model is pretty good. What's missing is the parameters, and I would argue the one major bottleneck between us and a truly personalized medicine is the parameterization of the very large models we have to deal with to be able to represent the differences between the different patients and the many different drug targets which the drugs which we want to model take into account, address. Those models are obviously therefore pretty large. This is one auto-generated image of our larger model. And I'm just showing you one result which seems to indicate that we're on the right track to be able to solve this parameterization model, which is not trivial because obviously we are dealing with parameter spaces of thousands of, tens of thousands of parameters which are non-trivial problem computationally. So I just show you one result in which we very early on, by a much simpler approach, have been able to treat a woman with metastatic malignant, or propose a treatment for a woman with metastatic malignant melanoma based on a, as I said, very simple model using a Monte Carlo approach for the, to overcome the need for the parameters. And you can see this patient 9, which is the one where you, the row which has this circle blue square, seemed in the modeling to react to one particular drug, which actually has not been used in cancer therapy, but has been used in treating rheumatoid arthritis. The left one of the two is the rheumatoid arthritis drug. The right one is MEK1, MEK2 inhibitor, which is in between approved for treatment and is was expected to work in a sense, but hadn't been approved yet. And the treatment success in the modeling, we in this case indicate by the blue color representing the concentration of MYC as a surrogate marker of cell division. At least a large part of cell division does involve increases in the MYC concentration. So the patient couldn't tolerate the standard treatment anymore because of side effects, and therefore the patient tried the rheumatoid arthritis drug, which kept the multiple metastases stable and shrinking for a long time till resistant metastases arose, which then also got analyzed, and it turns out that both the new model and the clinical practice showed that it was resistant to this drug. So those models are pragmatically restricted to the things we know are affecting the drug response. So, for example, for a patient receiving an oral drug, you would model the microbiome because that affects what actually enters the bloodstream, which forms the drug or drug metabolites enters the drug stream. You have pharmacogenomic processes in the liver predictable from the whole genome sequence or whole exome sequence of the patient. We have to model the tumor in as much of its complexity as we characterize. We are really limited by the input data. It's very hard to analyze multiple biopsies from the same tumor. And we should model the normal tissues because obviously it doesn't help us to find a drug which kills the tumor if it also affects the heart cells of the same patient. And in the case of using immunotherapy or taking into account the immune response to the tumor, we really should be able to model the individual immune system of the individual patient, which we are still quite a bit away from. So essentially what we are trying to do is just set up a situation in which the doctor, before treating the real patient, can test every possible treatment option, including combination drugs, which obviously are very much needed in a situation in which the tumors tend to escape any therapy because of heterogeneity and new mutations. First on the virtual patient, and then he and the patient can pick the most appropriate treatment for that individual patient. So again, you make your mistakes on a computer model of every individual patient. This has consequences also for drug development. It's a point has been raised how expensive drug development is. Therefore, that it is impossible to make a drug for every individual patient. First of all, we don't have to make drugs for individual patients. We just have to take the available drugs and test all possible drugs and drug combinations on the virtual patient. If we want to make new drugs, we can use the modeling to enormous advantage because it doesn't cost 50 million to run a virtual clinical trial. Virtual patients are insensitive against side effects of drugs, so we can basically take all the cancer patients which have been analyzed in ICGC or TCGA or in so many other projects and test their predicted response to a drug which has just been synthesized, so we know how which proteins it binds to with which dissociation constant, or in fact, if you have powerful enough docking programs available even before the drug or any drug has been synthesized. So in contrast to the current practice where developing a new drug costs somewhere between 4 to 12 billion dollars, if you divide the cost of developing, the pharma companies spend every year by the number of drugs which actually reach the market, we would be able to cut the cost by maybe 2 orders of magnitude or more. Those drugs not only cost much, they often also buy you very often only limited medical benefits, because again, most patients which are treated with the drugs don't respond. So my working hypothesis is most drugs which fail in development and are not developed would help specific subgroups of patients. We just have to know which ones. And that's something which we can expect to get, hopefully, from this type of models. Essentially, all models are wrong, but some are useful. What we aim for is useful models. We are obviously at an early stage. We are probably equivalent to the very early stages of weather forecasts in the middle of the last century. But even then, those numerical weather forecasts were useful. They were, for example, being used in the landing of in the Normandy, obviously with success. For this, we have been applying for a flagship project, 1 billion euros over 10 years, with the argument that without this technology, the EU would spend over 7 years, over the next framework program, 10 trillion on healthcare, 2 trillion on drugs to which the individual patients don't respond, probably have the equivalent of the population of Milan die of adverse drug reactions, and come 7 years closer to the implosion of the European healthcare system, and all that for the equivalent for the amount of money which Europe spends in 5 hours for its current healthcare systems. Unfortunately, this argument didn't seem to be strong enough for the referee panels. Those projects have been evaluated in. This goes way beyond cancer. It's a strategy which should be applied to all of medicine and all of prevention, and actually should also be applied to situations which are purely unrelated to medical applications. So they should give everybody more control over their own biology. So if you wanted to train for a marathon, you should be able to design the ideal diet and training schedule for being able to do so without getting off your sofa. That's people who have been participating in the slides I showed, the main people obviously. Marie-Laure Jasper's group in Max Planck is doing a lot of the work. She's also guiding some of the work at the Alakis Theranostics, at our startup company. The work would be impossible without close interaction with clinicians at the Charité and in other hospitals. And we also work closely with mathematicians, computer scientists to solve the many computational problems. For example, Jan Hasenauer at the Helmholtz Center in Munich, and Lucianito and Costin Ciostel in Romania. And that's some of the prizes which our startup got. So we are somewhat encouraged that this is maybe the way to go. Thanks.
Great vision of being able to predict. I know now that for even bacteria, linear programming models trying to guess the effects of even knockout of multiple genes at a time. Multiple effects have problem predicting how the system is going to work, and that's when we're talking about bacteria. So how far do you think we are from actually actualizing this vision?
I think we're pretty close. We have, as I said, we have had successes already with very simple models. You have to keep in mind that the response rate to drugs in clinical practice is not fantastically high. You have heard some numbers, so I would assume that with effects of $1 billion of funding for healthcare behind us, that we should be able to make models which are good enough to improve on that. The basic problem is, as I said, if we go to more complex models, we have to find the subset of the parameter space which gives us the right predictions on a test set. And that is something which requires resources, time, data, computation power. And unfortunately, that is something which we have only available through fairly small grants, which slow things down. The problem is that, for example, the solar cell industry got subsidized with 100 billion in to make their model, their the cells much more effective, much cheaper, and the same type of investment, not even on that scale, would really be needed to develop completely new technologies in medicine, being able to really change the way we treat cancer patients. So I think it's just a matter of resources and willpower to get very far. We are using, I've described this one case, we are using the models in clinical practice now, but we need to improve them a lot to make them more useful. It's, as I said, the main problem is the parameterization step.
Is there any data that shows the quantifiable, or that shows the advantage of these modeling approaches in a quantifiable way? I mean, I'm a little bit, this weather forecast of like approximation, you know. Of course, I look at the sky, I think it might rain, and I look at my appendix, it's at 50% of rain. I don't find this very helpful. I mean, we have to compare, we have some care for the patients. The doctors are giving a good, you know, they are making decisions, and they are often making good decisions.
They're also often making bad decisions.
We don't know. I don't
Yeah.
think it has been shown that these models
Okay.
have a specific advantage over the capital.
Yeah, ultimately we have to run clinical trials in which we compare the response rates from 500 patients getting treated according to the model with 500 standard patients. If 500 patients get up from their deathbed and go on vacation, I think every skeptic will believe that this is the way to go. So there's no problem. The problem is we have to develop the technology to that power, and if we only get the money to develop it, once we have run the clinical trial, which we can run once it has been developed, then it's hard without a time machine.
You know, some of these companies have been working on these approaches and building the databases for 10 years.
No, nothing.
Something delivered.
Okay, see, nothing of this form has been done. Okay, you cannot model without the data input. So all the talk about modeling was complete nonsense. You cannot model without having the data which we only have now. There hasn't been molecular models by any company. If you tell me one, I would be interested. There's a lot of talk about AI, deep learning, data-driven approaches. I'm skeptical. I think this is it's going to be very hard to train in a purely data-driven form neural networks to predict correctly on the training set how to treat patients. Basically, you need too many trainings data, and so you, in a sense, you almost have to repeat the evolution of man to be able to do it in a purely data-driven form. So none of that has happened. People have talked just as much as they have now talked for 15 years about personalized medicine without doing anything in that direction. There has been a lot of overselling. You just have to really discriminate what the technology can deliver at which point in time. This is not possible just by listening to buzzwords coming from companies or from pharma.
Yeah, you said producing models are only as good as the data are produced. Now there are, and you also said previously that an organism, human or not, is not the same as well. I mean, take the two things. And you said it makes the organism even easier because they don't change so much. But I think this is also a problem because we simply don't have the data. We don't have the data why development proceeds in a robust way, despite all the toxicity of the cell, chemical reactions in the cell. And so as long as we don't have them, they can put them in any models. And when then the tumor cell decides to abandon this way of remaining robust, we wouldn't even know. We wouldn't have the data to put them in. And so I don't really know how, I mean, the data are anything but comprehensive because basic research still doesn't know this mechanism.
We need comprehensive data for prediction successes of 99.9%. We don't need comprehensive data for doing better than the clinic of today, is my conviction. And I think that is simply because we have spent a lot of money already on basic research. And I would argue that if this wasn't enough to make models which are better than the current clinical practice, then we should have spent some money for public transportation or something useful. So I think it is, I'm convinced that basic research is useful, and my conclusion is based on this. I think in the next generation, we will have a more effective form of basic research, which is partly model-driven. Ultimately, you can, in research, you can only disprove. And so ultimately, what we will be able to do after the initial steps is formulate everything we know in models, take the models which we make to treat cancer patients, compare the results of the predictions to the real results, and thereby eliminate models and improve models, improve parameters. So those models are automatically self-learning, simply because we keep eliminating, we keep disproving potential models. So whatever remains becomes more and more likely to be true. I think basically, I think we are, the simple models will already predict the simple biomarkers. For example, the RAS effect, the effects of the of many of the mutations which are being used as biomarkers.
Thanks a lot for a very inspiring talk. In continuation with Professor Loeb's question, first of all, I would like to agree with the fact that maybe we're spending our money wrong. And as someone who's working in genetics, perhaps I'm showing my own life that maybe whole genome sequencing can seem with great expenditure and not so hard results. Maybe it's the wrong way. in terms of partial penetrance. So even if you know the genotype, you can't exactly know through the data. And in terms of exposure, simply drinking a glass of oranges can completely change the way that you react to a drug. And this is
Sure.
also something that should come into play with the type of prediction.
Yeah, you should be able to model the effect of grapefruit juice on the drugs which you take. If there's a clear mechanism there, or if you can deduce clear correlations, then you can put it into the model. In fact, ultimately the models will probably become hybrid models with strong mechanistic components and AI type parts wherever we don't have mechanisms. And that is something which many environmental exposures, we just don't have the mechanistic models, but we might be able to put them in by tricks like that. Anything you can measure on a patient which can be linked to the model is useful. So obviously those models should contain all the information, molecular as far as we can, ideally spatially resolved, sensor information, image information. Ultimately, we will be limited only by what we can afford to do with the patient, rather than with the technology how to analyze the samples.
And do you have a mechanism to integrate these kinds of things into your model?
For example, if you expand to heart disease, you would obviously use sensors to measure the physiology. You would link the physiological effects to the molecular effects happening in the heart cells. So you have to be able to link the components together, either by mechanisms, which we would have to explore in, for example, how the heart cells react to the pressure in the environment, or by AI type analysis, which then can be encapsulated as an object, in a sense, in a larger model. So anything which gives you correct predictions is useful. So I don't want to beat any of the AI techniques if they are useful, if they prove to make the right predictions on a test set. You know, all those techniques are fantastic on reproducing the training set. The problem comes in when you try to make predictions on the test set. Just reproducing the training set is not extremely helpful.